So my name is Seleipiri, and I'm going to be talking about using Assembloid systems for as a starting material for brain organoid-on-a-chip systems. So my first question from the poll, which I'm curious to see what people came up with, is when you hear the word assembloid, what comes to mind? So one definition is that these are the next generation, or arguably the next generation of brain organoids that combine multiple cell lineages in 3D. So putting this in context of what we know already about how to generate brain organoid systems and what has been echoed throughout the other presentations is that you derive them from these HIPSC clusters or embryoid bodies, and they can either undergo undirected or unguided differentiation or guided differentiation. So with unguided differentiation, you can, you're relying on the spontaneous morphogenesis and intrinsic differentiation capacities within these HIPSC clusters to form tissues mimicking the developing brain. Whereas with the guided differentiation, you're delivering external factors or external patterning factors to direct the stem cells towards a particular lineage. So with assembloid technology, you're kind of moving one step further with this guided differentiation, where you're trying to fuse either multiple brain region-specific organoids, or you're fusing multiple cell types within the organoids to better recapitulate what's happening in vivo. So this is a fairly new approach that's being used for brain organoids and has a lot of potential, but there is still a lack of integration of this with brain organoid platforms. So I'm giving an overview of brain organoid and chip platforms and what they've been used for, or kind of questions they've been used in addressing, particularly in the organoid generation process from the Chinhua Chin's lab. You can see that they've developed platforms for large-scale production of these organoids, as well as for looking at various disease modeling. So they tried to look at how nicotine exposure affects brain development in vitro. In other labs, they've also used it for modeling interstitial flow to see if they can recapitulate how fluid flow occurs around organs in vitro. And also something that I found interesting is using perfusion culture to reduce necrotic core formation during the culture process, which is a huge advantage of microfluidic systems. So besides these applications, one can envision that microfluidic platforms do allow for controlled better control of the culture process, and there are a lot of applications that these systems can be used for in future years. But I'm curious to see what the audience thinks are the perceived limitations of these pre-existing systems for brain organoids. So from my perspective, I think the limitations of this system lie on the starting material. So it's a consequence of what you're putting into them that gives your final product. So I'm going to be talking about two limitations of brain organoids that are used for these microfluidic platforms. And two of those limitations would be reproducibility and restricted cellular diversity. So kind of what has already been echoed in the other slides, but there's still a challenge of reproducibly generating the same number and types of cells in this organoid systems, which makes it really hard to do quantitative or comparative studies with these samples, particularly in the context of restricted cellular diversity. Because of the differentiation protocols and more biased towards the neuroectodermal lineage, you end up with a lot of neurons and a few glial cells, but there's still a lack of reproducibility in generating these cell types over time. So I'm going to talk about how assembloid systems can help in addressing these limitations. I'm not giving an exhaustive list of what the potential of the systems, but these are just a few examples to get us started on thinking or talking about how these assembloid systems could be useful. So particularly in the context of reproducibility, there's a lot of labs that have shown that by differentiating cerebral organoids towards the dorsal or ventral lineage, you can fuse them together to look at long distance interneuronal migration, because you can tag one of the cells in the organoids and look at the dynamics of the migratory process. People have gone as far to look at how drugs can affect this migratory process and better recapitulate what's seen in vitro, as opposed to trying to use a cerebral organoid system that may not fully represent all these cell types. And also in the context of restricted cellular diversity, there are a lot of approaches involving incorporating the cell types that are not present in the brain organoids, particularly microglia or astrocytes. And in one paper looking at even vascularizing the systems by incorporating endothelial cells into the brain organoids. So what's interesting is that they've been able to recapitulate some interesting cell interactions. For example, with the microglia incorporated organoids, they were able to observe the change in microglial morphology from this more quiescent state where it's in the ramified processes to when it moves to an amoeboid state when the brain organoid is injured or activated. And people have used these approaches to incorporate these very diverse cell types and further help in our understanding of how these cellular interactions occur during development. So although assembloid systems are useful for understanding these developmental processes, I think there's still a lot of work that needs to be done to fully utilize the potentials of the system. So I'm thinking that some synergy between the organoid-on-chip development and organoid differentiation protocols needs to happen, particularly in addressing certain challenges of the assembloid system. So one challenge would be ensuring that you have a defined cell polarity during the organoid fusion process. So with the organoid system, we still need to increase the robustness of the differentiation protocols. The assembloid systems are still relying on this guided differentiation of stem cell assembly. So being able to show that we can generate the same cell types from batch to batch is really important. And from the organoid-on-a-chip perspective, utilizing microfluidic approaches for spatial patterning would also be useful in ensuring that we can have these defined cell polarities during organoid fusion. So I'm going to give some examples of approaches that have been used so far in addressing these two solutions that I'm proposing, but I'm also curious to hear what the audience thinks in terms of addressing this challenge. So for increasing the robustness of differentiation protocols, there are a lot of labs that are currently showing that their differentiation protocol can result in similar cell types or similar diversity in cell types from organoid to organoid, at least with their protocols. And there are a few papers in literature that have shown that. And from a more synthetic biology approach, people have shown that by incorporating HIPSCs that can release sonic hedgehog morphogens over time within the brain organoid, they can induce a cell polarity within the organoid and better control the differentiation of cells over time. And from a microfluidics perspective, using well-established methods for generating these spatial gradients in vitro will also prove useful. So this is, I'm highlighting two more recent papers, but this is a well-established method in the field for creating chemical gradients and then looking at how that affects stem cell patterning in vitro. I'm going to end on this slide, which also highlights another challenge is how do we maintain these multiple cell types while culturing them in vitro? For the organoid systems, I'm thinking we would need to narrow down the key components for cell culture, particularly figuring out what growth factors would be necessary for culturing these multiple cell types. And for the organoid-on-a-chip side, I'm thinking we would need to develop approaches for doing multi-organoid-on-a-chip, not only to look at how these fused organoids are interacting with each other, but also looking at how brain organoid systems can interact with other organoid systems in the body. And that concludes my presentation. I'm willing to take any questions.
Wow, thank you very much. Does anybody have any questions for, I guess we could even include Onar if he's still on the call, since we didn't really get to ask him any questions. Any questions for either of the presenters?
So Pri, this is Roger. Very nice presentation. I was really interested, especially in your future directions.
Right.
Have you thought about how you would put organoids on a chip? That's something that we're thinking about and thinking about some with Ron Weiss. So I'd be curious to hear what your thoughts are.
So I actually found a few approaches in literature, not specifically for brain organoids, but at least for different organoid types. So I'm thinking more of like having them in their separate systems and then fluidically connecting them might be the first approach. Yeah. But the main challenge would be figuring out what the media component would be to perfuse through the entire system. But yeah, that was one of the ideas I was thinking of.
We're developing a system which might be of use to you, where you could have a different on top of the different organoids, and then also have all the organoids within the same gel, so that if you could vascularize them, which is the other thing we're trying to do, that potentially that vasculature could actually connect one organoid to another. So something to think about.
Okay.
I don't know if it'll work or not, but at least we got some ideas on how to try it.
Okay.
Great. Are there any additional questions?
Yeah, I guess I had a little bit more of a comment too. I really liked your perspective on this. It's kind of like a very new area of research with organoids, and yeah, I really like your point about how you can establish spatial gradients for axes in organoids, and it seems like it could go really well with a hydrogel if someone wanted to increase the scalability while still having an established axis. So yeah, I guess I don't know if you know of anyone who's working on that.
So I'm not aware of anyone using hydrogels, but there are approaches for culturing the. Okay, so there are two. I guess I have two responses to that. There are people that are using hydrogels for culturing organoids, so particularly pushing towards using synthetic hydrogels in order to avoid the use of Matrigel, which is animal derived. And then there are also other approaches, not necessarily with organoids, that allow you to spatially pattern your system. You can either add growth factors to the hydrogel matrix, or you can do photo patterning of the hydrogel in order to release specific growth factors at specific times. So there are definitely things that you can play around with in terms of using hydrogel systems. You can even combine that with the microfluidics-based platform to have better control of that. But I haven't seen a particular example in literature. At least I'm not aware of it.
It's cool though that you could combine them.
Yeah.
Very cool. And with that, I would like to share something with all of you momentarily. So we tried something today. I don't know if anybody can see this. I don't know what anybody is looking at this moment.
Yes, we can, Kaylee.
Oh, good. All right. So we wanted to try to do some poll questions today. And it's my first time using Poll Everything, so we're still trying to work out the kinks. I'm finding appropriate questions and ones that aren't appropriate as far as style. But these were the responses that we received today. For example, when you were asked what you thought of when you heard the word assembloid, these were the answers that were submitted. I mean, we can see organoids at the very top here in the largest letters. Some of the additional questions included perceived limitations of the MPS systems. The answers were everything from lack of blood supply, how did we define and compare functionality, reproducibility, scalability. There seemed to be a theme with these answers. And then likewise, we asked all of you, what's the greatest challenge facing the field right now? And almost 60% of the respondents chose reproducibility.